Hello. In this video, we will see adenylyl cyclase CAMP pathway of intracellular signaling. Let's get started. In the video on G protein coupled receptors, we have seen that two important types of G proteins are GS and GI. Adenylyl cyclase is affected by these two proteins. GS stimulates adenylyl cyclase and thereby stimulates the pathway. GI, on the other hand, inhibits the pathway. Generally speaking, stimulation of GS-coupled receptors lead to an increase in the activity of the cell, and stimulation of the GI-coupled receptor leads to a decrease in the activity. First, let's see pathway involving GS. GS protein consists of alpha S, beta and gamma subunits. Under resting state, the trimer of these subunits is associated with receptor and alpha subunit is bound to GDP. The binding of a ligand with the receptor causes conformational changes in the receptor and G protein that leads to release of GDP and binding of GTP. This in turn causes dissociation of alpha subunit and beta gamma complex. The alpha subunit travels along the membrane and goes to adenylyl cyclase. Adenylyl cyclase is a membrane-bound enzyme facing the cytosol. Alpha S subunit stimulates this enzyme. The enzyme then converts ATPs into cyclic AMPs. So the concentration of CAMP in the cell increases. Increased CAMP concentration leads to activation of CAMP-dependent protein kinase A. pKa phosphorylates different transport proteins, metabolic enzymes, transcription factors, or structural proteins. The phosphorylation modulates the activity of these proteins, which in turn affects various cellular functions. As example, let's see what happens in myocardial cells. They contain beta-1 receptors, which is a GS protein coupled receptor. Adrenaline stimulates these receptors and activates adenylyl cyclase pathway. This eventually leads to phosphorylation of proteins that sequester calcium into the sarcoplasmic reticulum. This ultimately causes an increase in the contractility of cardiac myocytes. Another example, liver cells. They contain glucagon receptors, which are also GS protein coupled receptors. Their activation by glucagon activates this pathway and causes phosphorylation of key enzymes for glycogenolysis. This ultimately leads to release of glucose by the liver. Now let's talk about termination of